Firstly, I'd like to thank the organizing committee for this opportunity. Hi everyone, my name is Nikhil George, and I'm a PhD candidate at the University of Waterloo in Canada. And today I'll be talking about virus-host interactions in a municipal landfill and how they include non-specific viruses, hyper-targeted viral populations, and interviral conflicts. For the past few years, the dominant virion morphology we've seen a lot of is this, SARS-CoV-2 from the family Coronaviridae. The reality is viral diversity comes in all different shapes and sizes. I'm specifically interested in prokaryotic viruses, as these viruses that infect bacteria and archaea are the most abundant biological entities on the planet. Yet despite this, I describe them as at least historically poorly characterized. And I say historically because it is recent metagenomic studies that have led the discovery of a multitude of viruses from a variety of environments. This altogether suggests we've been technologically limited in our ability to study these viruses in natural environments, but if we use the right tools and we look in the right places, we might uncover quite a bit. Beginning first with the tools, in our lab we use metagenomics. We go to a system of interest, collect a sample, extract DNA from that sample, and sequence this DNA to get these short fragments called reads. These can then be assembled into contigs, and the contigs can be genome binned, in an attempt to recreate the genomes that existed in the microbial community we originally sampled. Essentially, it tells us which bacteria and archaea are present in our system. The next thing is, where did we choose to apply these tools? For that, we go to my favorite quote, which is that one person's trash is another person's treasure. And I say this because my supervisor, Laura, really likes to study trash, specifically in municipal landfills. Our system for this work is the Southern Ontario landfill, from which we have two temporal samples, one collected in 2016 and one collected in 2017. So we have our bacteria and archaea from the genome binning I mentioned. To annotate the virus, we took our assembled contigs and we put them through vir sorter and vibrant, which annotate viruses as contigs that have an enrichment in viral or viral-like genetic features and a depletion in cellular genetic features. The next thing we needed to do is predict the hosts of the viruses we identified. To do this, we leveraged the natural biology of the CRISPR-Cas system, whereby if a virus tries to infect a bacterium or an archaeal cell but fails to do so for whatever reason, if that cell has a CRISPR-Cas system, it can cut up pieces of the virus's DNA and store it within its own genome in what is known as a CRISPR array, as a CRISPR spacer. It can then use this immunity as a molecular memory when confronted with a virus from the same population, so that the host can now mount the appropriate immune response to defend itself. It's kind of like how vaccination works for us. For our workflow, if we can match CRISPR spacers from host CRISPR arrays to viral genomes, we can predict that that host has been infected or can be infected by that viral population. And with this information, we can create our virus host networks, one of which is depicted here. One of the most interesting things we came across in this network was that some viruses were very strongly targeted by host CRISPR-Cas machinery. A representative virus is shown over here with its annotations up top, and the targeting profiles of different hosts to this viral element shown below. We see a strong degree of targeting to the tail protein encoding gene and hypothetical coding regions we weren't able to annotate with current reference databases. The next thing we found very interesting is we identified putative cross-phylum infecting bacteriophages. We think of bacteriophages as very specific entities, but our work among current research has shown that viruses can potentially infect across broad taxonomic orders, suggesting that these viruses are not as specific in natural environments compared to in culture or in the laboratory. The last thing I want to point out is some viruses can encode their own CRISPR-Cas systems and CRISPR arrays, which they use in interviral conflicts, essentially in competitions against other viruses. We identified 190 such viral elements in our data that encode CRISPR systems, and one of these CRISPR arrays is predicted to be the largest virally encoded CRISPR array identified to date. So, in summary, this understudied engineered environment houses unique virus-host connections and interactions that include non-specific viruses and hyper-targeted viral populations, and the observation of interviral conflicts shows the potential for CRISPR immunity-based super-infection exclusion. That brings me to the end of my presentation. I'd like to acknowledge my lab. I'd like to acknowledge the organizing committee for the opportunity to be here and the following funding sources. Thank you for your time.